Hello everyone. Today we are going to discuss structure of skin. Starting with the area of skin, that is 2 meter square, and it has got weight of about 4 to 5 kgs. Now, layers of skin. We have epidermis, which is composed of stratified squamous epithelium. It is made up of keratinocytes. This layer is followed by dermis, which is composed of connective tissue, collagen fibers, and blood vessels. This is further followed by hypodermis, also known as subcutaneous fat, and here we have lobules and septa. Lobules are composed of adipocytes and septa is made up of fibrous tissue. Right? So now, yes, very important question will come from this particular figure. Here we have layers of epidermis, and the topmost layer is called stratum corneum. It is made up of corneocytes. The second layer is stratum lucidum, and this layer is particularly present over palms and soles. Very important question, as this layer is present only over this area. Now, stratum granulosum. It is composed of keratohyaline granules, thus giving its name. It has got another important body known as lamellar body. Below it we have stratum spinosum, also known as prickle cell layer. It is known as spinosum because of the spinous processes because of desmosomes. Below it we have stratum basale, also known as stratum germinativum. The important question that comes from this particular topic is the layers that you have to enumerate from like top to bottom. Topmost is corneum, lucidum, then we have granulosum, spinosum and basale. Very important question. Now, what particularly happens when we move from basal layer to topmost layer? In stratum basale, we have tall columnar cells with nucleus in the center, and when we go to top, that is in stratum corneum, the cells becomes flattened and non-nucleated or anucleate. Right? So we have columnar cells in the bottom and flattened cells at the top in stratum corneum. And this particular process of cells to mature from basal to corneum takes about 28 days. Very, very important MCQ question. And this is known as epidermal transit time. Right? So, if this time decreases to approximately 3 days, as it is seen in psoriasis, the cells which were supposed to become non-nucleated remain nucleated, and this nucleated stratum corneum layer, which is seen in psoriasis, this particular process is known as parakeratosis. Okay, so this parakeratosis is a very, very important MCQ question that is seen in psoriasis, right? So if stratum corneum is thickened, this is known as hyperkeratosis. If there is thickening of stratum granulosum, this is known as hypergranulosis. And thickening of stratum spinosum is known as acanthosis. Very important questions. Few words about another important topic, that is epidermal melanin unit. So we have non-keratinocyte cells as well in skin, one of which is melanocyte, and one melanocyte gives melanosomes to 36 keratinocyte. So this 1:36 ratio is one epidermal melanin unit. Very, very important MCQ question. One must read this. Now, starting with another topic, intercellular junctions. Here we have a very important question on desmosomes. So, what are desmosomes? These are intercellular junctions which are particularly present in stratum spinosum. Because of these structures, we have polyhedral cells in stratum spinosum, and these connections give particular morphology to the cells. And if these connections are cut, as in one autoimmune disease, pemphigus, where antibodies are formed against desmogleins which are present in desmosomes, the cells change the morphology and they become circular, leading to blister formation. This whole process is known as acantholysis. Very important question. Now, we have another important topic on dermoepidermal junction, which is also known as basement membrane zone. We have total 4 layers of this dermoepidermal junction. First of all, we have hemidesmosomes. Second layer is lamina lucida. Third is lamina densa. And fourth is sublamina densa. So we are asked about layers of dermoepidermal junction from top to bottom and corresponding molecular structures. Right? So from top to bottom, what we have is tonofilaments composed of keratin 5 and 14. Then we have BP antigen 230, 180. Then laminin 5 in lamina densa, and anchoring filaments type 7 collagen in sublamina densa. So the question which is asked is the layers from top to bottom. So we have keratin 5, 14, BP antigen 230, 180, followed by integrin layer in lamina lucida, and laminin 5 followed by type 7 collagen. in anchoring filaments. Very important question. So, when these structures, these molecular structures are targeted, for example, BP antigen 230 and 180, we see a condition known as bullous pemphigoid, and type 7 collagen is targeted in epidermolysis bullosa acquisita. Another important question. So now, starting with dermis. Dermis is divided into two parts, papillary dermis and reticular dermis. Papillary dermis is composed of 1/10 and reticular dermis is about 9 to 10. And papillary dermis is composed of loose connective tissue and reticular of dense connective tissue. And the cells which are present in dermis are fibroblasts, macrophages, and we have lymphocytes. Now, the connective tissue of dermis is composed of collagen fibers, 70%. And we have type 1 collagen fibers. These are the most common type of collagen in skin, followed by type 3 collagen. Important question again. So these cover almost all the important questions that are being asked on basic dermatology. Thank you so much, everyone. See you next time. Thank you.